Imagine if you lived in a world where genetic diseases have been completely wiped out, and you could wave around a pair of biological scissors to cut and change the parts of your DNA you don't like. If given the chance, would you? One day in the near future, you might have to choose between yes and no. A cutting-edge gene editing technology called CRISPR has allowed researchers to add, remove, or alter genetic materials at particular locations in the DNA. The development of this technology has caused lots of excitement because with it, humanity has gained the power to engineer its own evolution by making genetic changes that will be passed down through future generations. DNA, also known as deoxyribonucleic acid, is made up of bases that are linked in a structure that we call double helix. The specific arrangement of the set bases codes determine who we are, and are responsible for everything from how we look to how we think. Every now and then, minute errors in the matching up of those four bases lead to what's known as mutations, which, although occasionally lead to characteristics better suited for survival, most likely lead to problems later on. In CRISPR, scientists first identify the bit of mutated DNA they want to edit, then make a simplified copy of the mutated DNA. This guide copy helps the enzyme, a biological catalyst, to recognize the mutant DNA once it has been inserted into the cell, enabling the enzymes to grow to two pincer-like structures, and literally snip the mutated bit, just like scissors. We can either remove it totally or change it. CRISPR will have a huge impact on how we treat diseases that are considered incurable today, because we can simply take out the bad parts of ourselves that make us sick. So, if CRISPR is so great, why aren't we rolling it out en masse right now? For one, scientists have cautioned against something called germline editing. This refers to editing done to cells responsible for reproduction, the sperm and egg. Mutations in these cells can be passed on to future generations, so any errors in editing could be passed to them, worsening any future problems CRISPR tries to correct. Besides the safety issues that could arise, bioethicists are also concerned about where we would draw the line. What if we could change a gene for the sake of higher intelligence? What if we could code for traits that are deemed desirable? Genetic enhancement and therapy is still debated hotly today, as some may believe that we shouldn't be interfering with who we are as human beings. They argue that it becomes a slippery slope. Once we begin to enhance ourselves, we might not know where to stop. CRISPR is a revolutionary technology that could very well change the way we, as a society, develop, and how we plan our futures. But we must be careful in how and when we use it.